All right. Hello, everyone. Welcome to Brandeis Mathematical Biology Seminar. Today we have Professor Jay. He's assistant professor at Department of Mathematical and Statistical Sciences at the University of Alberta. His research interest is motion of small things in microscale biological systems. Today, he will tell us about microscale fluid properties of nucleoplasm. Thank you, Jay.
Thanks for the introduction. Let me share my screen here, I guess. See if I can mess this up. Nope. Everybody see that?
Yes.
Okay. All right. Right, as the title says, I'm interested in modeling how the nucleus organizes itself. So the nucleus is a weird place, right? There are no plasma membranes inside the nucleus to organize subcompartments, to restrict some biomolecules that shouldn't mix with other things, maybe dangerous reactions. This, after all, is where all of our DNA is kept. So no membranes so outside the cell, you have membranes, organelles that organize things, but inside of the nucleus you don't. Nevertheless, things are highly organized in the nucleus, and not only that, but it self-organizes very dynamically, very quickly, as it goes between different stages of the cell cycle. So we're very interested in how this happens, like how does this self-organize spontaneously, and how is it maintained over time? Does it require energy to maintain this organization without compartmentalization? All right, so we have this overriding notion, right? When you have molecules mixing in a liquid, and you have Brownian motion, then over time entropy is going to want to just spread everything out so that everything is evenly concentrated. And so we generally think, energy is required in order to counteract that. Coming from the neuroscience space, I've done some modeling on the single neuron levels. I always think about ion channels and how important this idea is for the function of neurons. And the idea being you can have a higher concentration of stuff, ions, on one side, and then a lower concentration on the other. And then if you open up these channels over the membrane, stuff comes flooding through. So, and you need pumps, right, to push ions across against the gradient to create this ion concentration difference that you can use to promote action potentials and other things. So membranes are important, right? So how can we function? How can the inside of the nucleus function without them? So I think that the idea of a membrane is on the mind of biologists who are thinking about this. So that's important to remember. It's like in people's imagination, they want to find something that acts like a membrane that isn't a membrane, right? I'm not sure such a thing exists, but okay, so alongside all of this is the DNA itself, right? And not just DNA, but other polymers like RNA that also are involved in this self-organization. There are lots of, I guess, competing hypotheses about how the self-organization occurs in the nucleus itself. And that's a bit of a chicken and egg conflict situation, I guess, in terms of like what establishes these separated regions, whatever you want to call them. We call them maybe condensates. So item C here, liquid-liquid phase separation, is something that I've done some work on in the past and something I'm going to tell you about today. But not everybody believes that liquid-liquid phase separation, whatever that really means, is happening in the nucleus, and that it's some other thing. But I argue like something has to kick it off. There has to be an egg somewhere to at least initiate this phase separation, or these regions that are distinct in some way from the surrounding. So it could come from like literal sequence of DNA, but that's really not clear that these whatever regions that we're looking at are distinct or have DNA that is localized along the chromosome sequence. So it's not clear that that can be an explanation for how these are created. All right. So cellular liquid-liquid phase separation has been a pretty hot topic in the last 10 or so years. And people are proposing the concept of a membraneless organelle, right, inside of the nucleus or inside of a cell. I think it's a dangerous way of describing these things. So sure, you have stuff that's like swept into like a little pile of leaves, but that doesn't mean that it behaves the same way as if it were wrapped in a membrane. All right, so what is liquid-liquid phase separation? The example that we all give at this point is like oil and water, right? You have hydrophobic, hydrophilic interactions, and they separate, and ultimately all the oil and all the water will completely separate into two not many droplets, but just two phases. And you need something like what's the term? There's something that can stabilize droplets. Yeah, I can't think of the term. I can't remember the term right now, but or maybe another question too is what even is a liquid? I think that people are starting to rethink that. We want to describe things that are happening inside the nucleus on the micron scale, using the language of like materials, right? This is a liquid, therefore this is how it behaves. This is an elastic, or maybe something even in between, a viscoelastic fluid. So there's various theories for describing these. And I think the nucleus especially is really bending those definitions, and you know, I think we need to develop a new theory to explain the nucleus as a material, or however we plan to imagine it. And so the relevant idea for a liquid here is that the individual molecules are doing some sort of a random walk. They're mixing, right? And then the liquid itself is deformable, and all the other sort of continuum mechanics definitions that we assign to a liquid. But that's important when you're arguing about whether or not like some observed compartment within the nucleus is a liquid-liquid phase separation. So how would you even test whether or not it's a liquid to begin with? So one thing that we do, an important experiment for this space is FRAP. I mean, this is a very well-known experiment that's been around a long time, important for a lot of things, but especially here. So I think this is one of the experiments that I'm going to really focus on today. So if you've never encountered this before, the concept is very appealing to applied mathematicians, right? Because you can see it's like a solution of a PDE, right? You have some fluorescent dye that you're looking at, and the brightness is proportional to the concentration of this molecule, and then you blast it with a laser, a special laser, which can really can be very accurate and bleaches the fluorescent molecules, just destroys the fluorescent molecules, so the molecule itself is otherwise completely unaffected. It just no longer emits light, right? So chemically, it's exactly the same. It's just that now you've created these two visually distinct concentrations, one dark and one bright. So what do you watch? You watch the solution to the diffusion equation. You watch them remix, and then you sort of time how long it takes for them to mix, and then however long that takes is an indication of the diffusivity of these molecules. So the diffusivity is just, think of it as basically the variance of the increments of a Brownian motion of one of these molecules. Or you can think of it as something like a time scale for the solution to the diffusion equation to reach equilibrium, to reach the constant steady state solution. But most of the theory for this focuses on the idea of an infinite background, right? And so an interesting question is how do things change when you're inside a tiny nucleus and you have liquid phase separating droplets around? So that's something I'll come back to towards the end of the talk. Okay, so here's my main beef with thinking about these phase separated droplets as like membraneless organelles, or I guess I could say that here's one of the main differences between a condensate and something like an organelle that's wrapped in a membrane, right? And there's the phenomenon as well known, it's called Ostwald ripening. That is, if you have two droplets that are nearby, right, the bigger one is going to absorb the molecules of the smaller one until the bigger one just like completely drains and the smaller one just vanishes, right? So that's why, you know, when you have oil and water, there's just one big oil droplet, right, at the end when you hit equilibrium. You can't have stable little droplets everywhere without some kind of energy input. Okay, so a lot of people are really interested in how chemistry would stabilize droplets in the nucleus, because they are stable. So Ostwald ripening tells us something else, though. Molecules that are concentrated into these droplets are perfectly capable of doing Brownian motion outside of the droplet. So if we're in the context of the nucleus, this would be like the surrounding would be like the nucleoplasm, right? So one of these molecules of protein can pass from one droplet to another, right? So there's no barrier that is restricting the movement of molecules between these compartments, right? Okay, so here's a FRAP experiment. So here's the before picture, and then I think, let's see, right up there, you can see where the bleach happens. And then several droplets are bleached at the same time. Okay, so the top is the yellow, it's the combined signal from the red and the green. And so it's not really important at this stage what the difference between red and green is. This molecule called TDP43, which I'm going to explain in a little bit. But for right now, the take-home message is, okay, we bleach, and then we just wait and watch what happens. And molecules from the unbleached droplets are moving through the nucleoplasm and replacing the bleached molecules in those bleached condensates, right? So take-home message again, molecules are free to mix between these compartments. These organelles do not restrict that, okay? Which that would be a function for an organelle wrapped in a membrane, right? Okay, so here's a problem that we worked on. I guess it's been a year or two now. So this was a fun project that followed on some just basic modeling work that I did with Kelsey Gager with Kelsey Gager as a postdoc. Well, now she's at the University of Ottawa as an assistant professor, but then she was a postdoc. And she was working with Amy Gladfelter at UNC and Greg Forrest at UNC. And we were just doing some basic liquid phase separation type modeling on some related problems. So we were contacted based on a paper that we wrote by these individuals, Ying Yu and Don Cleveland at UCSD. Our work, we noticed that if we mix chemistry with the concept of phase separation, we could get patterning, you could say, or organization inside of the condensate itself. So we noticed that we could get these annular shells. That's why they contacted us, because they said we have this experiment of TDP43, this protein involved in neurodegenerative diseases, and we're wondering if you can explain our experiments, explain these annular shells. So you have to look at this picture and think like a biologist. It's very intriguing and tempting to think of some kind of a barrier like a membrane, right? When you look at this picture of an annular shell. So I think that it's important to remember that's kind of the idea that was going on in the background as we were trying to figure this out. All right, so what does TDP43 do? That's actually not really relevant. It's involved in pre-mRNA maturation. What's relevant and why people studying it are studying it is because it's involved in neurodegenerative disease, right? So it interacts with RNA, and it forms these condensates in the nucleus. And in the disease models, it's localized into disease-causing plaques, right? All right, so it dimerizes. So two TDP43 molecules will come together and form a dimer. And when they're dimerized, they can phase separate. And they also interact with RNA. Those are the relevant details right now. Okay, so here is I guess this is an electron, this is electron microscopy. So you can really see these annular regions, very distinct. They're very dark here. So again, intriguing to think of these as membraneless organelles, right? So there's some sort of structure here maybe, right, which is trying to confine whatever's on the interior. Let's see what else we have here. Here's a picture, here's a video that really convinced me that these are liquid droplets. These are liquid condensates. I'm looking at these and they are merging. And when they merge, they reorganize into these spherical, right, with the annular organization also re-emerging. So for me, liquids do that, right? I don't know how else to explain that, right? I don't know what caused them to separate and organize in the first place. Like I said, there's always this chicken and egg dilemma in this space, but I think these droplets are definitely liquids, right? I don't know what other explanation there could be here.
Jake, could you just say again this kind of annular structure? That's not really typical for Oswald ripening, is it?
Oswald,
so
you just mean liquid phase separation?
Well, yeah, this kind of annular.
Yeah, that's not typical. No.
Where
we found it in our simulations, but it was just a totally hypothetical thing. I actually went into the literature and looked around to see if I could find it anywhere else. And I don't know. I mean, so what is liquid phase? The physics of it. In the 60s, people came up with it. To mix better buckets of paint or something, right? It's it's an equilibrium sort of theory, and so it's difficult, and I think that's part of the challenge and the resistance that people have to it is taking that and then applying it to something that's going on inside of a living cell. So I guess maybe once you have it in a living cell, it's not unbelievable that you would see some crazy things, some weird new phenomenon that the people really hadn't thought about with this simple theory. Yeah, I'm not aware of any other instances of this annular organization though.
Jay, I have another question related. Yeah. So I was wondering if there was anything specific inside the annular structure, or if it's the same kind of material inside and out.
Well, Jonathan, that's going to be like the punchline of the story is finding out what's on the inside, right? Yeah, that's the question. I think that the picture that we had at the time, these being like membrane structures, they were also interested in whether there was some elastic properties, you know, or even some liquid crystal properties to the outside shell. But we actually weren't thinking about the possibility that what's inside of these things is any different from what's outside, like that it was just nucleoplasm in there, right? Because nothing that they had stained or looked for showed up as being distinct on the inside versus the outside, including the electron microscopy. So, all right. Why am I showing this again? Yeah, so TDP43 mixes between between these condensates. So we already made that point. Okay. So they're looking around at different kinds of different proteins that they can label, and just looking at how they're organized in the nucleus together with these TDP43 droplets. So TDP43 may be red in these images, or they may be green, so it kind of switches back and forth. So just be prepared for that. But here they're red, right? And GFP is just GFP, right? Nothing special about that. But what's clear, I think, is that GFP is essentially uniformly distributed in the nucleus, including inside of these droplets. So it's mixing everywhere, no problem, right? All right, so here's some other proteins. I don't know what these are, HNRNP, some sort of a RNA protein thing. And they also mix freely throughout the nucleus, right? Including the droplets. So there are these like dark spots, the nucleosomes, those are something else. The nucleoplasm, though, and the droplets, they seem to be able to mix just fine in there. Okay. So here is RNA, right? Plenty of RNA inside of the nucleus. And kind of interesting, TDP43 interacts with, the dimer interacts with RNA, it binds to it. It's involved in mRNA maturation, and yet they don't like to mix together. They like to be apart from each other. So what you see here is TDP40, this time in green, okay, is forming the annular droplets, and then the RNA is excluded from those droplets, right? So it's freely mixing in the nucleoplasm, but doesn't want to go inside the droplets. And there's an arrow here. He was really interested in whatever that was. I don't know what that is. Probably some accident, but okay. So the questions that Ying and Don asked Kelsey and I were, you know, how these droplets organize and assemble. What does it suggest about their function in wild type? And what does it say about their role in disease? And how are they disassembled, and how is their size maintained? There's some notion here that ATP would be required right, for these droplets to be stabilized. So that's the energy input that's required here. All right, so we're really just going to focus on this first question now. How do annular droplets organize and assemble? So this, again, it was based on some previous work that we had done, modeling. Actually, we were modeling liquid phase separation in the cytoplasm and not the nucleus in these fungal cells, Ashbia. And I'm going to show you guys Ashbia a little bit towards the end of this talk, so I'll come back to that. So the way that we modeled this phase separating system is a very common way of modeling this phenomenon, especially in the biology, liquid phase separation and cell biology sort of community, is just use Cahn-Hilliard equations. So you postulate a free energy, and then you look at sort of the gradient flows in that energy. So you can define these PDEs in the usual way using the functional derivative. And you get something that looks and acts a lot like a reaction diffusion equation, except now for the volume fraction of the phase that you're interested in, and there could be more than one, you have what's usually a fourth order operator, right? Instead of the typical Laplacian. Yeah, I mean, you need something to get these cool patterns, right? You have to have a more exotic equation. And there's some interesting links, some work that my colleague Thomas Hillen is involved in. There's some interesting links to non-local PDEs here. Okay. So that's our goal. Explain how TDP-43 organizes into these annular droplets. And can we explain it with a Cahn-Hilliard mechanism? So we were just taking our model, the numerical methods that we had developed for it, some pseudospectral methods to solve those PDEs, and just seeing if we could, put the chemistry into the model and see if we could get annular, because remember the TDP-43 is dimerizing, and then there's the interaction with the RNA. And so could we tune that chemistry in such a way that we get annular droplets? That was our question. So we can code these parameters. We can encode like how well each of these species likes to mix with the others, with these parameters in the free energy, which just think of them as interaction potentials. The higher the number, the less that they like to mix with each other. So this first one is the interaction potential between the dimer P2 and the RNA R, right? And so they don't like to mix. So that one gets a 4.25. So of course it's only the relative difference. It's an energy, so it's always the relative difference, right? That matters, not the absolute number. And then we have like the dimer likes to phase separate from the nucleoplasm S, right? The solvent. And RNA is perfectly fine mixing inside of the solvent. So it doesn't phase separate on its own, the RNA. So these are the basic details that we pulled just from the experiments that we were shown. And this is kind of what we would get, right? So on the left column is RNA at different snapshots of time going down. So initially it's at top, and then it's evolving as you move down. Of course, everything, because of Ostwald ripening, everything gets ultimately confined in just one droplet, right? So TDP-43 phase separated just fine. RNA was excluded, just like we saw in those experiments, but there's no annular organization to the red droplets on the right. So we couldn't really make that happen. So we decided to put a new player into the model, which was some unknown thing that's inside of the annular shells. So instead of thinking of that as just being like the solvent, right, we got some unknown proteins, RNA, some maybe some combination of the two. So really all we did was we just gave it an interaction potential, which was marginal, like in the middle. So the idea is that it's not strongly repelling the dimer. It likes to mix with it a little bit, right? But that's a Y here. The one in red is the one to note. And then the interaction between Y and the RNA, right? So RNA is repelled from it just as much as it's repelled from the TDP-43 dimer. And importantly, it has to want to phase separate on its own as well. All right. So then if we add this new player with this marginal interaction mixing potential, we could get annular TDP-43 droplets. Let's see, yeah. Takes a while for this to kick off. But... Okay. So that's what we brought to Ying and Don. We said, go find whatever it is that's inside of these droplets, which was an easy thing for us to ask, but Ying, you spent months searching, doing some pretty heroic work, probably very difficult, and ended up getting this very exciting find that there's this HSP70. It's a chaperone protein, which is really interesting because it probably does have this very marginal, that's a bad way to describe it. The way that it interacts with the protein TDP-43 is probably very weak, like rapid binding sorts of interactions that could lead to a marginal mixing potential, I think. Okay, so then the story goes on. So Kelsey and I, of course, we did the modeling part of this paper. The story goes on, and so the idea is that HSP70, these chaperone proteins, they regulate TDP-43 activity and then spatially organize into these droplets, these multi-component droplets. And then TDP-43 forms an annular shell, which potentially restricts access to ATP or whatever. When ATP levels drop, and the ability of HSP70 to maintain these droplets is degraded through some downstream mechanism. They form aggregates, right? So if we solve neurodegenerative disease, probably a small step in that direction, right? This is a long process, but this is one of an interesting early stage of that process. All right, so my work on trying to understand the organization of the nucleus has moved on. I'm now working with my colleagues here at the University of Alberta. Tharana is a PhD student working with Gerda de Vries and I. And then we are working with our biology collaborators, Alan Underhill and Michael Hensel, who've been working in this space for a long time. And so is Gerda. So this paper, which I really love, is Gerda's paper from 2003, where they were modeling an experiment and then doing data analysis with that model in what I think is an exciting way. I like doing this stuff. And so they were looking at context for FRAP, right? So I mentioned this before. The theory behind doing inference with a FRAP experiment, getting a diffusivity value, right? That theory is all based on, being in an infinite ocean of some, right, unbounded space, and then bleaching a small region, right, and then waiting for it to remix. So there's no context like the outer boundary, a confining domain, and there certainly isn't any kind of context in terms of condensates, right? Liquid phase separated condensates. So the concept here is that you have binding and unbinding to some substrate. This could be like chromatin, for example. You have this, you know, wound up DNA with these histones on them, and these definitely are a substrate for proteins to bind and unbind very rapidly. So then you can have like multiple compartments, these phase separated condensates, and then you can ask, all right, what are these parameters, D1, D2, and then the binding and unbinding rates. And that's something that they were actually able to infer from the FRAP video data, which I think is pretty cool. So that's kind of where we were starting, and we wanted to try and say something about FRAP with liquid phase separation, right? Because that's one of the very important experiments in this space, but nobody has really updated the theory behind FRAP to work with liquid phase separated droplets, and also not inside of the nucleus. So here's a curious experiment. I know this video is it's looping really fast. I don't know why it plays so fast. Right. So can you see the cursor over? Okay, good. So here, I love this experiment. Is a partially bleached. Now these are chromocenters. Chromocenters of another protein called KMT5C. This is a totally separate, you know, thing from the earlier where it was TDP43. I always get them confused. Too many of these weird protein names to remember. Anyway, so KMT5C, it organizes inside of what are called chromocenters, and this is a partial bleaching of that chromocenter, right? And what you can see is the typical idea of just mixing, right? So some random motion is occurring there so that whatever unbleached molecules remain in that chromocenter are spreading out and to fill in that hole, right? And the bleached molecules are also mixing, right? So everything should be getting a little bit dimmer. But then, of course, you also have mixing between these chromocenters, which is easy to forget about. Okay, so what else is in here? It's kind of hard to see, but there's a fully bleached droplet right there at the center. Okay, so then we're watching to see how fast it refills when everything is bleached inside of a chromocenter, and that is very slow. Okay, so I don't have another movie for this for context, but a very similarly sized molecule, CBX something, I can't remember, 40, I think. It matches, so it also localizes to these chromocenters, and it matches the behavior of the partially bleached, right? So the size of this molecule is such that, you know, its diffusivity is going to cause it to spread out at about the same time scale, 60 seconds. So we're talking like a minute or two, right? It's gonna spread out. But unlike the KMT5C, this other molecule will also draw new unbleached molecules from the surrounding chromocenters very quickly, on the order of like a minute or two. Whereas the fully bleached droplet here does not recover. And they can change that just by changing the chemistry of this molecule, KMT5C. They can regulate these two behaviors, right? The time to remix or to recover when you partially bleach versus when you fully bleach, right? So this requires, I think, theory to put FRAP in the context. So that's ongoing work. Okay, so one more quick I don't know, how much time do I have? I still have time, right? Do we end at 11? Okay. I gotta cruise through this then. All right, so this is with collaborator Amy Gladfelter, UNC, and graduate student Grace McLaughlin. So I'm also interested not just in the organization inside of a nucleus, but how nuclei organize, right? So this is a cell where we have many, many nuclei. It's a syncytia. So how does this cell self-organize all of its nuclei? They do this very elaborate dance where they're using active motion, molecular motors, microtubule transport, and they're dividing as they move through their cell cycle. Asynchronously, we don't know how they maintain asynchronous divisions. That's one mystery. And we also don't know how they're organizing, because we don't really know why they're organizing in the way that they are. Like, we don't know what the function is, I guess.
So what we're seeing here is just one cell with multiple nuclei that are moving?
That's right. This is all one cell, Ashbia gossypi, and these bright spots are all nuclei. There's like hundreds and hundreds of nuclei inside of a single cell.
And the shape of the cell is not this star, right? It's much larger. This is just a pattern inside the cell, or?
The branched, so these form, I mean, we have this idea of the fungus forming these tubular structures, right? In soil and some potential interaction with trees, right? This is what we're seeing here. It's a single cell. We're seeing most of it here. So it's like cut off, like where the base of this branched tree. So it sort of looks like a hairball, right?
Okay.
Yeah. So it's branching for the same reason, say, that a neuron would branch. It's trying to cover a lot of volume outside with not a lot of volume inside the cell. Does that make sense? It wants to minimize the amount of cytoplasm it's using, right? Because that's growth that requires energy. But it wants to go out and try to get food from as wide an area as it can. Yeah. So do these nuclei compete with each other? Do they cooperate with each other? Why the very deliberate spacing between the nuclei? And they also do something very elaborate. You would think that as they divide, they stay as like nearest neighbors in some kind of a network, like a graph. But they very deliberately shuffle themselves and mix that graph up. And so right now, what we're trying to do is track these nuclei so that we can connect them to models and try and figure out how could we do the basic data inference to say how synchronized two nuclei are in terms of their cell cycle, right? That's something we would like to be able to do, quantify that. And we could maybe connect that to a model like the theory of coupled oscillators. So we can talk about phase response curves of a cell cycle oscillator, right? So here's an example of Drosophila. Okay, so I just wanted to show, like, here's synchronous nuclear divisions in a syncytia. This is the standard typical situation. The cell cycle proteins are just mixing really fast outside of, all the synthesis has to happen outside of the nucleus, right? So the cell cycle proteins are definitely outside the nucleus, and they're small, so they diffuse quickly. And so that couples the cell cycle, the oscillatory chemical reactions that are underlying the cell cycle. So they couple and they synchronize, right? So why is that not happening here? They're very close to each other, right? These are just like a micron or two apart, and the cell cycle proteins are small and diffuse, definitely fast enough to couple the cell cycles there. So we don't know how that works. Here's another video of them growing, right? So then there's this idea of slow growth on top of everything else. We need, in order to answer some of these questions about divisions, we need to get a video long enough to capture multiple divisions, but they're moving all the time, right? And sometimes they move really fast with motor-driven motion, but other times they're just kind of like going with the flow. So just to get this video, Grace had to really just finely tune over months to be able to capture this, to get a very long recording. And now we've just finished tracking it. Yeah, so this is ongoing work as well. So that's all I have to share with you guys today. Thanks for inviting me. I gotta thank my funding support. Yeah.
Thank you, Jay.
Thanks.
Okay. So I mean, I can start with one question maybe. So if I understand well, the theory for the generation of annular domains, it's in fact, you just create kind of circular domains, but then you make them compete with each other. And so the one that has more, that really doesn't want to mix, creates a circular domain, and then the rest cannot make another circular domain. So just an annulus.
Yeah, I think that the key is, so why don't they just form their own droplets?
Right.
That's actually the typical situation. So why would they cohabitate in the same droplet and then organize within it?
Right.
So that it's important that the mixing be sort of in between, like this marginal, so that they co-localize the same droplet, right?
Yeah.
So sort of like to mix with each other, but.
So precisely my question was really, did you make a bifurcation diagram? If you put 4.25 on whichever was inside, you're gonna have just droplets, right? If you just
Exactly.
one, yeah. When does it separate? And
you should
going closer from 4 and then C.
I don't think we did that for this paper. I would have to go back to look to our earlier paper, though. Kelsey definitely did these parameter, these bifurcation diagrams. Yeah, not for the same model as this one, but yeah, there's very careful, cataloging of the different sorts of patterns that you can see.
Mm-hmm. I was also thinking, does it depend on diffusion of molecules? And you would think that maybe if they diffuse fast, you may compete against the formation of clusters, stuff like that.
Yeah, that's interesting.
That's
so we definitely, so we included in the model a phase-dependent diffusion with the idea that the droplets themselves are far more viscous than the surrounding nucleoplasm. So we did include some of these effects, but they weren't necessary for the organizational effects. Yeah, so I don't remember if we did the sort of the bifurcation diagram with the diffusivities. I think we're mostly focusing on the chemistry. The chemistry seems to be what living systems use to go beyond, Brownian motion and the equilibrium situation. I guess you could characterize it that way.
right. Oh, very nice work.
I also wanted to ask, like in the syncytial cell, the multinucleated one, do you see any clusters of all of those nuclei? Are they kind of uniformly distributed through this cell structure?
Yeah, we want to use the word uniformly distributed, but that it's almost not enough, right? Because they're like very evenly distributed. Like they somehow know, and this is work that Amy's done in the past, they somehow know that the nucleus to the number of nuclei to the volume, the local volume of the cell. They know about this ratio, right? So they're trying to maintain like this very constant density, if you want to call it that, right?
So they're almost like regularly spaced.
Yeah, right, exactly. not perfectly regular, but definitely non-random.
Isn't that just, I mean, could you have that just with repulsion?
Oh, yeah. So there's definitely some kind of
confinement.
repulsion. There's some work. So they you know, there's microtubule dynamics and the polymerization, depolymerization of the microtubules is a thing. And then the motors, you know, they're pulling along those microtubules. And so it's like having an oar, and it's like pushing against the other nuclei, and it's pushing against the cell wall. And, you know, it's very complicated. Who knows how so, I mean, I guess that would be the microscale explanation for how they're moving around and pushing off objects that are sort of far away.
And you said they're dividing asynchronously. Does that mean that there's a whole distribution of sizes of these nuclei?
That's right. So they're essentially at random stages along their cell cycle, but no one has ever really quantified that. Like, what even is synchronized or not synchronized, and so there's this notion that if they did an experiment where if a nucleus was transported past another nucleus, right? So they were briefly right next to each other, that their cell cycle would be a little bit correlated afterward, right? So they had, you know, they used whatever methods to try and quantify that, but that's a difficult question. Just, you know, to say how coupled or how synchronized are two nuclei. Could you look at this thing, one of these videos, and say, right, these are not synchronized? I mean, I guess that's what we do, right? Because it's definitely different than the Drosophila picture. But, and for me, I think the theory of coupled oscillators does that very nicely. It's the literal difference in phase, right? That if that's 0, then they're synchronized, and if it's not, they're not. So that's what I would like to be able to. I would like to be able to infer that, right, from tracking not only their position but their lineage, right, when they divide. So if their two daughter cells move away from each other, and we track how, you know, they're dividing, where they're at in their cell cycle, we can get some idea of how that evolves over time. But you can't decouple the tracking from getting the lineage. That makes it kind of difficult because they move. It's almost like they're deliberately trying to evade us watching them. I mean, they do this thing where they get close and they like tumble around each other, right? Like that game. What is it? The game. When people on the street are trying to steal your money, where the shell, is it shells? Yeah, you guys know what I mean. It's like they're trying to do that.
One quick biology question.
Okay.
Do you have any data, or going back to annuli? What happens biologically if the size changes? Is there any biological phenotype?
The size of the egg?
Because seems like cells actively maintains the size, not too small, not too big, right?
Oh, you mean if the cell is bigger, then the nucleus is also bigger? What happens to these, like in general for like condensates, you mean?
Oh no, I mean not sure if you have any biological data, but like is there any mutant or something? Each annuli becomes bigger than wild type or smaller than wild type, and then what happens to the cells?
Yeah, so there's definitely like a distribution of sizes, and it's something you might miss, and I think that's the question you're asking, like, why are they the size that they are? What determines that? It seems to be somewhat stable. Maybe there's actually a better picture here. Let's see if I can find it. That's not a bad picture, but I was thinking of this one. You can see a lot of them there, right? Little ones, big ones. All right, so there's some distribution But there seems to be like an upper bound on the size. And so what determines that? Is it just the merging? It can't be, right? Because then they would just merge into like one huge droplet. And now not only the merging, but the Ostwald ripen, right? So something has to be counteracting. That's where the ATP comes in, right? Or energy. So we know that these HNP proteins interact with ATP. So there are other forms of energy, is all I'm saying. But there has to be some energy input, some chemistry that leads to this. A lot of people are interested in that exact question. Like how does that maintain? What determines that size? We don't know. Yeah, that's a good question.
It's a stupid idea, but if you overload the cell with ATP, do you get smaller annuli?
Yeah, that's a good question. Can you perturb the ATP levels and then see what the effect is like? I think I always propose these experiments, and then the biologist was like, "Oh, come on, you can't." I mean, that would be cool, though.
Yeah, that would be cool.
I'll ask Ying next time I talk to him.
Yeah, I'm a biologist, by the way. Maybe I'm on the stupid side of biologist. Anyway, yeah. Do you have any other questions?
I think Simon has one.
Oh. Hey, how come I don't see it? Oh, got it. I allowed you to talk.
Thank you. Yeah, and thank you, Jay, for the presentation. I don't have like a specific question on the material, but I'm really curious to know like how did you prepare the slide, and look like you're not using Microsoft PowerPoint or Beamer, so it looks really interesting. So I don't know like how did you prepare these slides and how you presenting them? Thank you.
Good question. I wanted something in between like PowerPoint and Beamer. I want to be able to use LaTeX. And like if I change a color, I don't want to have to use LaTeX it on every single little snippet 500,000 times to make it, because you're always mixing different presentations together, right? You don't want to use the same style all the time. And I like playing a lot of videos, and I hate the way that PowerPoint does that too. And Beamer, like it won't do movies, right? Or it sort of does, but it's a big hassle. Yeah, so this is all just based on a web browser. It's called a Reveal. I have a thing on my GitHub to just like a really simple working example for using math and playing videos. But if you Google Reveal, slide presentation, then it'll take you to the to the main page. It's not my own creation. I'm just using it. And it's great because you can display anything that you can display in a web page, right? You can put into these presentations. So it's a little bit more work because you have to write HTML and JavaScript and whatever, but it's really not that different from Beamer and LaTeX. So
so I'm looking at a web page right now.
Yeah,
that's awesome. And then how do you spell review? Is it like just REVIEW?
Yeah. Reveal.js, because it's a JavaScript package.
Very cool. Yeah, thank you. I appreciate it.
Okay. So you can put interactive, JavaScript plots and things. Yeah, you could really go hog wild with it.
Yeah, I remember you have this simulation where you could change the parameters live. I told you that, I mean, but I was never able to implement it, in fact. But I have your Reveal thing. And it's beautiful.
Yeah, so JavaScript is a powerful, scripting language. It's like having MATLAB inside of your presentation slides, right? You could literally do a simulation right there in the slide. Sometimes that's bad though, because I do everything stochastic, right? So you have a random simulation. Sometimes they don't do what you want them to do.
You cannot pick the right example too.
Yeah, that's right. Do the right thing. Yeah.
Oh, that's so cool.
It's really great for movies because the movies start when you go to this slide and then they stop when you move away and then they remember where they're at and then they start again. It's really
Oh,
smooth.
wow.
Yeah. The transitions are very smooth and seamless. Like I hate the way the PowerPoint does them. Like you have to go through the entire transition one after the other, right? Where here you can just skip. Like if you hit the arrow key a bunch of times, it's just going to zoom through all the slides.
Yeah, it's beautiful.
Wow.
So it just like feels more natural, I think.
So they also like the pencil icon, the bottom, like can you also do annotation and
Yep. It doesn't work super well, but yeah, you can. Yeah.
and the icon right next to that, it should give you a list of slides.
You can even write like on movies, yeah. And then I don't know what this does. Oh yeah, I guess you can skip around to the different slides, yeah.
Thank you again. I appreciate it.
Thanks for asking. Yeah, I want to convince more people to use this, but
Great.
All right. If we don't have any more questions, let's thank Jay again.
Thanks, Jay.
thanks
you.
everyone. It was good to see you guys again.
Good to see you. Bye-bye.
Good to see you.
Yeah.
So for the rest of us, we'll have the seminar in person next week. So see you again.
See you. Bye-bye.
Bye.